Data storage. As time goes on, the amount of data we store is increasing exponentially. Consequently, we have to find more and more ways to store all of this data. Hard drives went from the size of a fridge, storing only 5 megabytes, to a matchbox, storing terabytes. As the amount of data increases, the size of the storage medium decreases, which reflects Moore's law, stating that the number of transistors in a CPU doubles roughly every 2 years. But is it possible to decrease this even further? Yes. What if we store data using nature's most efficient storage system, DNA, the programming language of our genetic code? The main advantages of using DNA is its extremely long shelf life and its compactness. 1 gram of DNA can store 215 petabytes of data, which is about 300 million CDs, which is incredible. First, let's look at DNA. DNA stands for deoxyribonucleic acid. It holds what is essentially our genetic blueprint. DNA is formed of 2 polynucleotides coiled into a double helix structure. Each polynucleotide is made of lots of nucleotides, containing a pentose sugar, a phosphate group, and a nitrogen-containing base. These nucleotides form phosphodiester bonds in condensation reactions, which are catalyzed by the enzyme DNA polymerase. The 2 strands run antiparallel, meaning in opposite directions, and are held together by hydrogen bonds. Let's now look into the bases mentioned earlier. DNA contains 4 bases: adenine, guanine, which are purines, and thymine and cytosine, which are pyrimidines. A complementary base pairs to T, and G to C. This pairing ensures that the diameter of the helix is constant, and is partly responsible for the thermal stability of DNA. To give a sense of perspective, about 4,022 nanometer transistors can fit across the width of a human hair. Now, how can DNA, which has the width of 11 times smaller than a transistor, be used to store data? Computers use base 2, ones and zeros. However, DNA has 4 bases, which means information stored in binary has to be converted to base 4, using A's, T's, C's, and G's. Therefore, theoretically, each nucleotide can hold 2 bits, but due to read and write errors, they hold a little less. This is done using a process called DNA fountain encoding, which is an algorithm using 3 main steps: pre-processing, Looby transform, and screening. The DNA oligonucleotides have extra tags added on them, which act like molecular markers, to help to reassemble the data in the correct order when reading. These are then synthesized. Each base is added one by one, going through stages of protection, base coupling, and oxidation. This forms an oligo pool. After the DNA is synthesized, it is dried and put in a container to be protected from humidity and light. How is the data read? The oligo pool is PCR amplified and sequenced. This helps to determine the precise order of nucleotides and generates a letter sequence which is then decoded into binary. Using the tags, the oligonucleotides are assembled into the correct order of the original data. As the DNA is read, it is destroyed. Therefore, when initially synthesizing the DNA, multiple copies are made for future use. What are the pros and cons of doing all of this? DNA has a much longer shelf life, is more durable, needs less maintenance, and is more compact than a normal hard disk drive. A DNA storage system also eradicates the need for expensive and high-maintenance data centers, subsequently reducing global warming as we save power. However, the drawbacks are that the process is currently very expensive and takes too much time to make. DNA fountain encoding can write about 100 bytes per second, whereas a modern hard disk drive can write hundreds of millions of bytes per second. Also, at the moment, the whole oligo pool has to be read, even only when a small part of it is needed. So, what now? This idea has already been tested. Nick Goldman and his team at Bioinformatics have stored a TXT file, an MP3, and a PDF using DNA. But because of the disadvantages mentioned, at the moment only government files or medical records are worth storing in this way. However, as time goes on, technology will undoubtedly advance, and DNA storage systems would eventually be able to be used in our everyday lives. Data can go from being stored in a warehouse to a walnut.